Hello, my friends, and welcome to our video lecture on topic D3.3, homeostasis. This is our additional higher level content. We're going to talk about the kidneys today. Our guiding questions continue to be, how are constant internal conditions maintained in humans, and what are the benefits to organisms for maintaining those constant internal conditions? Our objectives today are all about those kidneys. We're going to talk about how the roles of the kidneys include excretion and osmoregulation. We're going to define osmolarity, talk about the structure and the function of kidneys, and then nephrons, which are the functional units of kidneys. We're going to talk about osmoregulation as a negative feedback loop, and then we're going to talk about some variations in blood flow to organs like the kidney. And remember that homeostasis is all about maintaining some internal condition at a stable and constant level. We do this through the use of negative feedback loops. We have already talked about how pH of the blood, body temperature, and blood glucose concentrations are maintained through homeostasis and these feedback loops. Today we're going to look at blood osmotic concentrations. How much water are we keeping in the body? The two functions of the kidney that we're going to look at today are osmoregulation and excretion. This molecule is urea. Urea is a waste product that is produced in the breakdown of proteins. It is moderately toxic, and so we definitely want to get it out of our bodies. When our blood is filtered by the kidneys, one of the products that gets filtered out is urea. That urea is sent to the bladder, and then we excrete it in our urine. Osmoregulation includes a whole lot of hormone feedback loops. When we notice with our hypothalamus that our water levels in the blood are a little bit low, we secrete a hormone. We're going to talk about this more in a couple slides. That's going to allow us to keep more water in the kidneys, which is going to get our water levels back up. This is osmoregulation. Osmoregulation depends on this idea of osmolarity. Osmolarity is related to tonicity. We have already talked about the words hypertonic, isotonic, hypotonic. Remember that hyper is all about a high concentration. If I have a high concentration of solutes outside the cell is going to lose water. That loss of water is due to osmotic pressure. If I have a hypotonic solution, hypo rhymes with low, this low solute concentration outside the cell causes water to move into the cell. That movement, again, is all about osmotic pressure. Pressure is the tendency of a solvent, in this case the water, to move across a membrane due to those differences in concentration. So tonicity is kind of measuring the concentration, and then osmolarity is about moving the solvent because of some of those solute concentrations. Osmolarity is measured in osmoles per liter. We could also say osmoles per cubic decimeter because a cubic decimeter is the same thing as a liter. And here we're going to take a look at the structure of kidneys so that we can talk about some of the functions of these pieces and parts. We humans have 2 kidneys. Blood comes from the heart out of the aorta, the descending aorta. We have some branches that are going to go into the kidneys. We call those the renal for the kidney arteries because they're going away from the heart. Blood is going to circulate around the kidney, get filtered. It's going to get sent back into the circulatory system through the renal vein. The renal vein is going to dump the blood into the inferior vena cava, which is going to carry that blood back to the heart. The outer layer of the kidney is known as the renal cortex. The renal cortex is here. Renal for kidney, cortex for that outer layer. Just inside the renal cortex is the renal medulla. Remember, medulla in the middle. There are some other super cool structures inside the medulla, the papilla, the pyramids, the columns. We don't really need to know that. Just know that the medulla is in the middle. Just inside the medulla, now we have the pelvis. The pelvis is here. The pelvis is where the urine starts to collect as it's been separated from the blood. The pelvis is going to merge into the ureter. The ureter is going to carry the urine to the bladder and then to the urethra and then out of the body. And we're going to zoom in here on a single nephron. Nephrons are the functional units of kidneys, just like the alveoli are functional units of the lungs, or sarcomeres are the functional units of muscles. Nephrons, this is where the work of the kidney gets done. And again, they're pretty small, which is why we have to zoom in really closely. Each one of our kidneys, remember that we have 2 kidneys, each kidney has about 1.25 million nephrons, lots of nephrons doing lots of work. Blood is coming from the renal artery, lots of branches. We end up with some arterioles, little arteries. Those guys continue to branch into capillaries. Some of our capillaries form these glomeruli. A glomerulus is this ball of capillaries under crazy high pressure, and some of the fluidy parts of the blood are going to ooze out of the capillaries because those walls are so permeable. That fluid, that filtrate, gets captured by this super cool guy, Bowman's capsule. The fluid is then sent through this first tube. We call this the proximal convoluted tubule. Proximal because it is close to the glomerulus, convoluted because it's wiggly and jiggly, tubule because it's a baby tube. The proximal convoluted tubule is then going to merge into the loop of Henle. The loop is literally a loop. It goes down, it comes back up. The loop of Henle is then going to merge into the distal convoluted tubule. Still a baby tube, still wiggly and jiggly, but now we are far away from the glomerulus, hence distal instead of proximal. That distal convoluted tubule is going to then dump the now urine into the collecting tubule. Sometimes we call it the collecting duct. Same thing. That collecting duct is going to send that urine to the pelvis of the kidney. There it is going to get sent into the ureter, to the bladder, to the urethra, and then out of the body. Surrounding these tubes, the proximal convoluted tubule, the loop of Henle, the distal convoluted tubule, are all these amazing capillaries. We call these capillaries the peritubular capillary bed. Peritubular capillary bed. Peri because it is surrounding the tubes of the nephron. Peritubular capillary bed. Its job is to collect all the stuff that we don't actually want to secrete in the urine. We're going to zoom in again, my friends, and look at just the glomerulus and Bowman's capsule. The function of the glomerulus and Bowman's capsule is ultrafiltration. Not just filtration, my friends, but ultrafiltration. Blood flowing through the capillaries of the glomerulus is under really high pressure. Here we're zooming in again. Blood is flowing through the capillaries of the glomerulus. The walls of the glomerulus, the capillary walls, have lots of fenestrations. Those are windows, and that allows for some things to ooze out of the capillaries of the glomerulus. Not cells. Cells are too big. Not protein molecules. Proteins are too big. But water and salt and glucose and that urea molecule, these guys are all small enough. They are going to ooze out of the capillaries and then into Bowman's capsule. They become part of the filtrate, that filtrate. Surrounding the walls of the capillaries of the glomerulus are these super cool cells called podocytes. Again, the blood in the glomerulus is under crazy high pressure. And so we don't want the walls of the glomerulus to rupture under that pressure. We want them to be nice and strong and stable, but we don't want them to be too thick because then they're not filtering so great. Stuff is getting stuck inside. So these podocytes are going to wrap around the glomerulus capillaries to help support them, but they have all these super cool finger-like or foot-like projections, hence they're called podocytes. And these projections just help to hold the capillary structure while also allowing for there to be lots of spaces for all the filtrate to get out of the capillaries and into Bowman's capsule. We also have a basement membrane that helps to support the walls of the capillary. We end up with that filtrate. That filtrate is captured by Bowman's capsule. Bowman's capsule is not going to allow that filtrate to escape into the rest of the kidney. Instead, all that filtrate, the water, the sugars, the salts, the urea, is going to get sent this way to the proximal convoluted tubule. And again, Glomerulus is here, all those podocytes, the basement membrane, the fenestrations, to allow for ultrafiltration. The filtrate gets captured here by Bowman's capsule. Bowman's capsule is going to send that filtrate, the water, the sugars, the salts, the urea from the blood that got filtered through the proximal convoluted tubule. The stuff that didn't get filtered out, the red blood cells, the white blood cells, the proteins are going to stay in the capillaries and then end up going around our peritubular capillary bed. In the proximal convoluted tubule, we are going to reabsorb some of the things that we want to keep in the blood. Again, we've got some salt, that's sodium chloride, we have lots of water, we have some glucose, we have that urea. We actually want to keep the glucose. We do not want the glucose being lost in the urine, and so we are going to reabsorb with some active transport the glucose. We're going to pull the glucose out of the filtrate, put it into these cells of the proximal convoluted tubule so that we can keep it, because we want to keep our glucose because we need to make some ATP. A super amazing adaptation of the inside of the proximal convoluted tubule. It has these little projections on the cells. So here's a cell. Here are all those amazing mitochondria. We have these wavy lines on the cell membrane that's facing the inside of the proximal convoluted tubule. We call this the lumen. The lumen of the inside of the lumen is the inside of anything. So here in the lumen we have all of these little guys. We call them microvilli. The job of the microvilli is to increase the surface area. So we're not just collecting glucose here, 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 but we're collecting glucose here, 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 here. Oh my goodness, so much surface area to allow for more absorption of that glucose. That's the job of the proximal convoluted tubule. Proximal convoluted tubule. Next up on our path through the nephron is the loop of Henle. So again, the filtrate is going from Bowman's capsule through the proximal convoluted tubule into the loop of Henle. What's pretty cool, everything that we've looked at so far, Bowman's capsule, the glomerulus, the proximal convoluted tubule, these guys are all situated in the cortex of the kidney. That's that outer layer of the kidney. The loop of Henle dips down into the medulla. The medulla is super salty. It is a hypertonic environment, and that's going to lead to some osmolarity and the movement of water due to that hypertonic environment. How do we get the medulla to be hypertonic? Well, we have some active transport of sodium out of the ascending part of the loop of Henle. So the ascending loop, the part of the loop of Henle that goes up to the distal convoluted tubule, is going to actively transport sodium chloride out and into the medulla, and that makes the medulla super salty. And now I have a hypertonic environment in the medulla, and water in the descending loop of the loop of Henle is going to, via osmosis due to osmotic pressure, it is going to move out also into the medulla. Remember that surrounding the medulla we have our peritubular capillary bed. This water is going to get absorbed into the capillaries. The capillaries are going to transport that water back into the circulatory system through the renal vein and then into the vena cava. So we can grab the water out of the filtrate by making the environment of the medulla of the kidney super salty. We get that water, we put it back into the capillaries, we keep it into the circulatory system. The rest of our filtrate, the stuff that we don't want to keep, we got the glucose out here, we got the water out here, we got the salt out here. What we have left is extra stuff, water, and definitely urea. The water and the urea are going to go now into the collecting tube. The collecting tube, which we can also call the collecting duct, is going to send that urea and water, urine, into the pelvis of the kidney, to the ureter, to the bladder, to the urethra. But what if we are not drinking enough water? Well, then our hypothalamus is going to notice that we have a low water level, and the hypothalamus is going to make this hormone called anti-diuretic antidiuretic hormone. Antidiuretic hormone, also called ADH, antidiuretic hormone. The hypothalamus makes it, sends it to the posterior lobe of the pituitary gland. The pituitary gland is going to release it. The target of antidiuretic hormone is cells that make up the walls of the collecting tubule. This is showing one of those tiny little cells here making up the wall of the collecting tubule. The receptors for ADH are on the wall. ADH is the ligand. It's going to bind to the receptor. ADH can also be called vasopressin. We're just going to call it ADH. So ADH, vasopressin are going to bind to the receptor. That's going to set off a signal transduction pathway. We're going to have this beautiful secondary messenger cyclic AMP. We are going to transcribe the gene that is going to then end up with the translation of a protein that is called aquaporin. Aquaporins are these super cool protein channels that are embedded on the inner wall of the collecting tubule. Aquaporins are protein channels that are going to, you guessed it, collect some more water. And so water can move through the aquaporins out of the filtrate that's on the inside of the collecting tubule. That water is going to move out of the filtrate. That water is going to go into the walls of the collecting duct and then get sent back into the circulatory system so that we can keep it in our bodies and not excrete it in the urine. Now, if we have been drinking water the way that we should, we don't have that ADH being made. We're not turning on those genes, we're not adding more of those aquaporins, and then we're allowing lots of water to be excreted with the urea in the urine. And here's the same idea, just another more zoomed in look. Again, here's our ADH. That's that vasopressin word again. So ADH is going to bind to its receptor. We have our cyclic AMP, signal transduction pathway. We're going to activate some genes. We have transcription, translation. We are going to have some beautiful exocytosis of these vesicles that include our aquaporin proteins. We have more aquaporin proteins in the inner wall of the collecting tubule or collecting duct. Again, this is the lumen, so this is where the filtrate is moving. The aquaporins here will collect more water, pull it into the cells so that it's not lost in the urine. But if I'm drinking lots and lots of water, I don't need all those aquaporins pulling the water back into my body. And so instead of doing exocytosis to express those aquaporins on our cell membrane, we do some endocytosis and we pull them back in away from the walls of the collecting duct, and then the water doesn't get pulled in, but instead keeps going along its merry way to the ureter, to the bladder, and then through the urethra, out the body. This beautiful negative feedback loop. I don't have enough water. I make some ADH. I make some aquaporins. I keep more water. I have plenty of water. I don't make the ADH. I don't need to make more aquaporins. And then we're able to keep that water level just right. Also, drink water. An interesting aspect of the kidneys, because they are considered so vital to staying alive, is that regardless of whether we are at rest or working out big time, the amount of blood flowing through the kidneys stays about the same. 1 liter per minute when we're at rest, maybe a tiny little bit more when we are running around. Same is true of the brain. Brain is super important. It's going to keep a pretty consistent blood flow regardless of whether we are hanging out reading a book or running around. That's different for the heart, of course, because how are we going to get more blood around the body with the heart? So when we are at rest, we've got maybe 0.2, 0.25 liters per minute of blood flowing through the heart. When we are working out, we multiply that by 5 times as much blood flowing around. From 5 to 25, 5 times as much blood flowing around the heart every single minute when we're working out. Also, the muscles are going to have a big increase in circulation. We go from a 0.75 liters per minute to 20. Oh my goodness, so much more blood there in the muscles, of course, because they're doing a lot of work They need the glucose and the oxygen that is transported in the blood. We take blood away from the digestive system when we're working out. We tend to rest and digest. We don't run and digest, which is why sometimes if you try to work out on a full belly, you get some stomach cramps because you've got all this food sitting in there and no blood to take care of that food. So rest and digest, my friends, rest and digest. And on that note, we have accomplished all of our objectives. We talked about how the kidneys are in charge of excretion, especially of that urea, and osmoregulation, regulating the water levels in our body. Osmolarity is all about those differences in concentrations and how that leads to the movement of solvents, the water, due to those concentration differences. We talked about kidneys and nephrons and ADH and its amazing negative feedback loop. And then how the kidneys tend to have not a lot of variation in its blood flow. Other things like muscles and the heart have big changes to blood flow, depending on whether we are at rest or running around. Great work today.